Good afternoon. Welcome. My name is Pat Granahan, and I'm here with Chelsea Loughran. We're both from both Greenfield and Sachs here in Boston, Massachusetts, and have really welcomed this opportunity to speak to you all about CRISPR-Cas9. And as you can see, we're going to be looking at CRISPR-Cas9 and what the IP implications might be for this new technology, which really has been, really at the tips of many people's tongues scientifically in recent months. And we thought it would be a very interesting topic to discuss with you today. One thing I wanted to point out is that we have been working on this, looking at it since last fall. We've been relying on only publicly available information, so that the information that we do have is information that you can access either through our website ultimately or through many of the government websites. In addition, Chelsea and I are working on a paper which we hope to have done fairly soon, which we will be very happy to send to you when it's ready, and it will include the references and information, much of which you'll see today. And if there are questions, we hope you'll submit them, and then if there's time at the end of the presentation, we'd certainly be willing to, very happy to answer them, and if not, we'll find another way to get back to you personally. So this is Chelsea Loughran.
Thank you, Pat. As Pat mentioned, I'm a litigation associate at Wolf Greenfield. I've been following the CRISPR technology and the IP landscape for this technology for the past about 5 to 6 months. And I know we have a number of different types of people in the audience today. I think we have some attorneys, some scientists. So I'm hoping that the information that we provide here today will be of use to the broadest kind of audience that we have watching today. Just by way of just kind of a brief overview, what we're going to cover today. So I'm going to give a brief summary of the technology and why this technology has generated such enthusiasm in the field. I'll quickly run through kind of the benefits of this technology in comparison to existing genome editing technologies. We did an overview of the research publications, and by way of timeline are going to provide a summary of kind of who has been doing the research in this field, and that then segues into what the IP landscape looks like in terms of the people who have been doing the research and who are simultaneously pursuing IP rights on the CRISPR technology. Then we'll talk about some IP unknowns. I think one of the unique things about this technology is that we are following it kind of as it's happening. It is new. It is cutting edge. People are publishing on new developments. The IP is still largely unpublished, and so we're following developments as they occur in real time. So there are a lot of IP unknowns. We'll also talk about some of the challenges that we think may arise in terms of securing IP rights to some of this technology. And then we'll conclude and, as Pat said, take questions if there's time. So no exciting, revolutionary new technology would be complete without kind of being associated with a face. And kind of the late-breaking news is these are the two new faces of CRISPR. These are two monkeys who were generated by researchers in China using the CRISPR technology. These monkeys were created with 2 genetic mutations using the technology that we're going to talk about today. And so this was end of January 2014. There has been some excitement over this. Some people are appropriately skeptical about how, what this actually says, but the use of CRISPR in primates such as monkeys is kind of the new news in this technology. So if you signed up for this webinar, you've probably heard of the word CRISPR before, but taking a step back, CRISPR is an acronym. It stands for Clustered Regularly Interspaced Short Palindromic Repeats. What does that mean? So by way of history, this whole technology started, well, the beginning of this technology dates back to 1987, when a group in Japan was doing some research on bacteria and sequencing bacterial genomes, and noticed these interesting repeating sequences in bacterial genomes. And what they were noticing was a series of nucleotide sequences, about 20 to 30 base pairs in length, that were repeating themselves, that were interspersed with unique sequences. And at that time in 1987, that was acknowledged, it was noticed, but nobody really could attribute a biological function to what people were seeing as these repeats sequences. If you flash forward by about a decade, what ended up starting to happen was researchers had basically started to understand that the unique sequences in between the clustered repeat sequences, the CRISPR repeat sequences, actually corresponded with sequences found in phage. Phage are, basically invaders of bacteria. And we get to 2007, and researchers at Danisco, which is a division of DuPont, began to attribute a function to these unique sequences. And what was realized was that these CRISPR repeats and the unique phage sequences in between them were actually a form of bacterial immunity, a bacterial immune system to fight against phage. And so kind of at a high level, what happens is bacteria is exposed to a virus, a phage. The genomic material for this phage is incorporated into the bacterial CRISPR sequences, a long array of phage sequences separated by these CRISPR repeats. And it serves for the bacterial cell as a type of memory of prior exposure to phage. When that bacteria is subsequently exposed to phage again, it triggers a cascade whereby that phage genomic information is recognized within this long sequence where you have your phage sequence and then a repeating sequence and then phage sequence and then a repeating sequence. The particular phage sequence is broken off from this long sequence and becomes a homing mechanism for a protein, a nuclease, to target the phage DNA, the phage sequence, and to destroy it. So it's basically an identification and a search and destroy capability for bacteria. Well, this was interesting, and researchers at Danisco made good use of this information in order to start to create more robust types of bacteria, having been able to understand that these CRISPR sequences confer bacterial immunity. Again, fast forward 5 years, and we'll go through some of the publications associated with this. But what has come from the bacterial immunity functionality of CRISPR is now the ability to do genomic editing. And the concept from a high level is that you can create a guide RNA, a sequence of RNA that is homologous with pretty much any sequence in any genome for any species. By creating this guide RNA and associating it with a CRISPR-associated protein, this is Cas-9, this is the protein that actually does the DNA cutting, which has 2 active sites for double-stranded DNA cutting. You can use the Cas-9 guide RNA complex to target any DNA sequence and affect a double-stranded break. And so from a kind of the basic CRISPR mechanism is the ability to home in on any DNA sequence using a guide RNA and the special Cas9 protein and affect a double-stranded break. Subsequent research is now being done to modify this in order to create more subtle impacts on a target genome, either by repressing DNA function or overactivating DNA function. And subsequent research has also demonstrated that you could use this technology to do multiple editing of multiple loci in the genome, so that you can create multiple guide RNAs to target multiple sites of interest in the genome and get multiple DNA editing functionality. Perhaps more interesting than that is the ability and the speed by which this technology can be used. In order to create transgenic animals, for example, transgenic mice, all you really need is the sequence, the mRNA sequence for the Cas-9 protein, your guide RNA. You can inject it into a single cell embryo and into a mouse, and that mouse will give birth to your mutant. And researchers have shown that you can do a single genomic editing, multiple genomic editing. You can, along with simply doing DNA cutting, you can add a target sequence of interest and incorporate that into a mutant's genome, which obviously holds great potential for doing DNA repair and other therapeutic capabilities. So how does CRISPR, I mean, everybody's very enthusiastic about this technology, but I just want to go quickly over how this stacks up to prior genomic editing technologies and why people are so enthusiastic about this technology. This is a comparison between CRISPR and prior genomic editing technologies, specifically zinc finger nucleases and talons across a number of categories. To dive in, zinc fingers were basically, research on zinc fingers began in the mid-90s. They are used for genomic editing. It is a approximately 30 amino acid protein that is associated with 3 nucleotide bases, and you can build 3 protein modules. So this is 1 zinc finger, 2 zinc fingers, 3 zinc fingers that would be associated with 9 specific nucleic acids. You can associate these with an enzyme, and the way that zinc fingers work is that this enzyme will do its DNA cutting as a dimer, and so you have 1 specific module here that will locate and bind to your particular DNA target of interest, another module of zinc fingers here, and when they locate onto the correct DNA sequence, the enzymes align and will cause your double-stranded DNA break. This has been, very useful in terms of doing genomic editing, and again, you can supply some type of corrective or target DNA that can be incorporated into the genome using zinc fingers to do your DNA cutting and homologous recombination to insert your new DNA into the genome. The problem with zinc fingers is that it does involve building proteins, and so through the process of building proteins is complex. It takes time. It does take money, and in terms of the ability for zinc fingers to target any locus on the genome, there are certain limitations because each zinc finger does recognize 3 base pairs rather than a single base pair. And so there are limitations on regions of the genome that can be targeted efficiently. And there are IP issues. A lot of the intellectual property on zinc fingers is owned by Sangamo Biosciences that operates a proprietary platform called CompoZX. About a decade later, enter talons. Now, talons are an interesting genomic editing platform. First discovered as, TALEN stands for a transcription activator-like effector nucleases. These were proteins that invade plants, xanthomas. And the way that these proteins target DNA in plants is that they comprise long sequences of amino acids, 33 to 34 amino acids, all identical except for the amino acids at position 13 and 14. It was discovered that those amino acids code for a particular base, and when you have these long sequences of amino acids, You look at these two bases here, they code for thymine, for example. The next sequence may code for arginine, and so on and so on. And using these proteins, you have basically a way of coding for a specific DNA of interest. Again, you can associate these with a nuclease and do your kind of DNA homing, double-stranded break, homologous recombination, genomic editing. Talons had many advantages over zinc fingers. The one-to-one association between a particular sequence and a nucleotide was a benefit over zinc fingers. I like this one more.
I can see it.
And Talons, as a genomic editing platform, seem to be an improvement in terms of specificity, ability to target any sequence in the genome, and although off-target concerns were thought of early stages in this technology, those concerns are being addressed as research proceeds. So far, Talons appear not to have the same IP issues as zinc fingers. But again, it is protein design, which can involve, obviously, time and money. These are also large proteins, so you have these long sequences of amino acids, one after another, and there has been some speculation as to whether that might limit delivery options as a therapeutic. So now, getting to CRISPR, I think probably the most advantageous aspect of CRISPR is, instead of it being a protein-based genomic editing tool, it is a RNA-based genomic editing tool. And by that I mean the actual tool that you are using to identify and home in on your gene of interest is a sequence of RNA. In nature, the way that this works is you have basically two associated RNAs. A CRISPR RNA, which is associated with, in bacteria, would be associated with the phage sequence, and thus identifies the target gene of interest, and a precursor RNA that is involved in actually creating this CRISPR RNA from a long sequence of CRISPRs and spacers. In genomic editing terms, scientists are able to create artificially this type of structure through a single guide RNA, hairpin loop type sequence of nucleotides. And so you have your code for your target of interest here. You associate it with this Cas-9 protein, which is capable of doing the double-stranded break, and you can introduce this into a cell, and the CRISPR-Cas9 will identify the target gene of interest and introduce your double-stranded break. I think that most people have associated CRISPR as a much quicker, perhaps cheaper, and more accessible technology because of its simplicity. There have certainly been concerns about off-target effects, about using this technology and perhaps trying to identify and affect a double-stranded break at a particular location, but for some reason getting that effect at the wrong location. There have been a number of publications that have come out that have done research on how to improve specificity and minimize off-target effects. But in terms of the IP, I think the message is to stay tuned, and this is why we have the presentation here for you today. So who has been working on CRISPR? I put together this timeline. It kind of tracks through some of the seminal papers in the CRISPR technology. The first one, as I mentioned, was a March 2007 publication by Rodolphe Beringau and colleagues from Danisco, identifying the bacterial immunity functionality that was associated with these CRISPR sequences. Several years later, Emmanuelle Charpentier and her group started to characterize how the long CRISPR sequences in bacteria were broken up into guide RNAs, so that those CRISPR sequences could be used to identify a target of interest and destroy it. And in the case of bacteria, that was the mechanism of bacterial immunity. I believe that most people associate the publication on August 17th, 2012, by Emmanuelle Charpentier, Jennifer Doudna, and her postdoc Martin Ginnick as the first characterization of the CRISPR-Cas9 system for genomic editing purposes. And the conclusion of her paper was the identification of a DNA interference mechanism involving a dual RNA structure that directs Cas9 endonuclease to introduce site-specific double-stranded breaks in target DNA. So this, I think, is widely regarded as the first publication that said, hey, we could use this technology to artificially create our own guide RNA and use the capabilities of the Cas9 protein to do genomic editing. And from there, so many publications have come out that have described this technology for various applications, have further described the capabilities of this technology, and have begun to envision this technology as kind of a breakthrough platform for doing genetic therapies. In February 15th of 2013, Feng Zhang and his group at MIT and the Broad Institute reported on doing multiple targeting of DNA using several different guide RNAs. And so he reported on multiple guide sequences being encoded into a single CRISPR array to enable simultaneous editing of several sites. So this is a very easy and rapid way of doing multiple site genomic editing. In the same exact issue, George Church published the very next article in that issue. George Church, who is a researcher at Harvard, also published on doing multiple guide RNAs affecting multiplex editing of target loci. He was also able, using computer technology, to calculate the actual number of unique guide RNAs that would be able to target the human genome, and put that number as of February of last year, so as of a year ago, at about 190,000 unique guide RNAs that could target approximately 40% of human exons in the genome. I'm sure that number has gone up since then. And just so once again, I mean, this is George Church's publication, his kind of image of the CRISPR Cas9 complex. But again, it's very simple. It involves your guide RNA that encodes your target gene of interest. Right here, a Cas9 protein that has 2 active sites for double-stranded cutting. And when this guide RNA locates the DNA sequence of interest, Cas9 affects the double-stranded break. And then in May of 2013, Rodolphe Janish's lab, who out of the Whitehead, published on doing 5 different disruptions in 5 different genes using CRISPR-Cas technology. So that's kind of an overview of some of the players in the field who have been doing this research. There are many others, and it's incredible to see the amount of people who are using this technology in various different ways. Right now, I'd like to start to talk a little bit about what the intellectual property landscape for this technology looks like. And we're going to kind of start in a less obvious place, but going back to the original discovery of these CRISPR sequences having conferred bacterial immunity in bacterial cells. The research that was done at Danisco and Rodolphe Beringau's paper back in 2007. If you look at the intellectual property, you'll see that the DuPont group has secured intellectual property rights and is pursuing intellectual property applications similar to the work that was done and reported in that first paper. So this is one patent family. There is a issued patent issued to Rodolphe Beringau, and this intellectual property is largely directed to methods for enhancing bacteria, methods for typing bacteria, methods for enhancing the resistance that bacteria have against phages using the CRISPR technology. So for example, one of these claims, so you have one of these claims in an issued patent, method for typing lactobacillus species having a CRISPR region that comprises certain characteristics. And so in terms of the intellectual property for a genomic editing platform, this may not be all that relevant. These claims are directed to kind of a different use of CRISPR. But these patents do discuss CRISPR as a discovery, as a technology, and the DuPont Danisco group is still pursuing claims directed to different variations of CRISPR. And so claim 21 here, this is a claim in a published application. So this isn't an actual claim that anyone has received, but this is a claim that exists in a still pending application. Claims an isolated nucleic acid molecule selected from the group consisting of, and includes a number of sequences directed to CRISPR, so various CRISPR sequences. This claim was subject to a restriction requirement. The Patent Office said you can't claim all of these sequences, you need to pick one. And at the end of January of this year, the Danisco group elected sequence ID number 1. And so they're still pursuing claims directed to particular CRISPR sequences. Now, for those of you in the IP world, isolated nucleic acids, claims directed to isolated nucleic acids, could run into problems with being patentable in and of themselves. But right now, this claim is early in prosecution, and the Danisco group is pursuing a claim along these lines directed to a particular CRISPR sequence. On the other side, kind of two separate patent families, it's interesting to look at the early claims in some of these patent applications. So these are the claims as they were filed for a particular DuPont application. And you can see that these early claims are incredibly broad and seem to actually kind of give a broad description of what CRISPR and Cas could be capable of. So, for example, this claim 1, use of one or more Cas genes or proteins for modulating resistance in a cell against a target nucleic acid or a transcription product thereof. Similarly, 21, a nucleic acid sequence consisting essentially of at least one Cas gene and at least 2 CRISPR repeats. Now, these are the claims as filed. These I haven't issued. Nobody owns these claims. There's been a series of amendments. Claims have been withdrawn, and prosecution has gone in a different direction. But it's interesting to look at these original claims because they seem like they might be broad enough to cover CRISPR for genomic editing, or at least some portion of it. A nucleic acid sequence that consists essentially of a Cas gene and 2 CRISPR repeats, that actually might be what people are using in doing CRISPR for genomic editing purposes today. Again, nobody has these claims, but they were part of the application when it was originally filed. The other thing to note is that all of these applications teach what they teach, and so could potentially be, prior art to any later applications. But when you look at this claim as it exists today, as currently amended, it's directed more specifically to a method for modulating resistance in a cell against a target nucleic acid or transcription product thereof using Cas, or CRISPR and the Cas system. But looking at these applications, it's interesting to see some of the interaction with the patent office that these applications are facing, and we'll get into some of that as we go forward. There are 2 pending applications also to the Danisco group that discuss methods of generating CRISPR variants and how you might create increased resistance bacteria by tweaking the CRISPR sequences. Again, this is an original claim, and this is the claim as it exists now. There are claims directed to actually labeling bacteria so that you can identify which bacteria is resistant to which particular phage it may have interacted with. And so, for the most part, the DuPont-Danisco prosecution is directed to this type of use of CRISPR for enhancing bacterial resistance. And I think that makes sense given that DuPont is in the business of kind of food products, yogurt, things like that, where high resistance bacteria is valuable. Now, kind of taking a step in a different direction, I think this application is actually quite interesting. So Eric Sontheimer is out of Northwestern University. And it is interesting to note the date on this patent application. So this was filed in September of 2009 and published in March of 2010. And if you look at the original claims, so the title of this patent is Target DNA Interference with CRISPR RNA. And the original claims of his patent, and again, these aren't issued claims, these were the claims that he included in this patent application when it was filed, recite a method of inhibiting the function and/or presence of a target DNA sequence in a eukaryotic cell comprising administering a CRISPR RNA, which is the guide RNA in modern genomic editing terms, one or more Cas proteins, or nucleic sequences encoding one or more Cas proteins, wherein that CRISPR RNA hybridizes with a target DNA sequence and interferes with the function of that sequence. And that sounds a lot like what CRISPR is being used for in terms of genomic editing today. And these were claims that were filed back in 2000 and I believe 2009. The same with this one, a method for regulating gene transfer within a cell using or inhibiting horizontal gene transfer wherein CRISPR loci are configured within the DNA of a cell. And so these were the original claims of Sontheimer's application. If you look at Sontheimer's research, though, he took this in a different direction. He realized that you could use this CRISPR technology in actually to prevent nosocomial infections, the transmission of antibiotic resistance bacteria in hospitals. So, I mean, it goes in a much different direction than the genomic editing technology, but he basically, and so his claims kind of went in that direction as well. Claim 10, as it currently exists, recites a method of treating or preventing an infection comprising administering CRISPR RNA, the guide RNA, and one or more Cas proteins or nucleic acid sequences encoding those proteins to a subject infected by S. epidermis or at risk of this S. epidermis, wherein the CRISPR RNA hybridizes to your target sequence and interferes with the function or presence of that target DNA sequence. So his vision and his claims kind of went in the direction of using CRISPR to prevent staph infections and to minimize horizontal gene transfer between bacteria, so as to to kind of reduce the spread of highly resistant bacteria in, for example, hospitals. This claim went abandoned, and so is no longer pending. So this application didn't amount to any actual issued claims, but the fact that it was filed and it teaches and it seems to describe CRISPR and potentially CRISPR in terms of doing DNA interference or somehow inhibiting or interfering with DNA seems to be a very early description of CRISPR in the public domain. Also, a different group, and this is a group out of the University of Georgia, Rebecca Turns and Michael Turns. They have also been doing CRISPR work, and this is actually an issued patent, and it is issued under the title Prokaryotic RNA-like System and Methods of Use. And the claims, again, this patent issued in October of 2013. The claims of this patent are directed more specifically, or at least appear to be directed specifically to CRISPR sequence as they would be found in a microbe. And so they recite an isolated polynucleotide comprising at least 23 nucleotides, where that polynucleotide comprises a siRNA tag, and PSI stands for prokaryotic silencing RNA tag, and a guide sequence where this tag corresponds with nucleotides of a repeat from a CRISPR locus that are present in a microbe. So these are issued claims, and they recite kind of a polynucleotide sequence that you would find in some type of prokaryotic CRISPR system. So it's unclear whether this type of claim would cover just CRISPR in its kind of genomic editing sense in terms of a guide RNA and a sequence that corresponds to a CRISPR locus. But this is an issued patent. It was issued to Rebecca and Michael Turns, University of Georgia. And in addition to this issued patent, they also have a pending publication that is cited on their website. If you Google CRISPR and the University of Georgia, you get to their website, and it cites this application as available for license. Now, I think that this application is directed a bit more specifically to a different Cas protein, Cas-9. And Cas-9 is implicated in kind of creating your guide RNA, or creating the CRRNA that is used to identify your target DNA. So when you have that long stretch of CRISPR sequences and you have your short spacer sequences, Cas-9 is implicated in actually kind of cutting out the target DNA of interest and then providing that for targeting and double-stranded breaks. But this is a pending application that Georgia has cited on its website and indicates is available for license. And the Turns, Rebecca and Michael Turns, continue to do research on various applications and aspects of CRISPR. So the next one is, recall we talked about the kind of the paper by Jennifer Doudna and her postdoc Ginnick and Emmanuel Charpentier. The next patent is a published application by their group out of the University of California, Berkeley. It published in May of last year. And I believe that the Berkeley group was, in addition to kind of first publishing on the CRISPR system as a potential new genomic editing technology, was largely involved in kind of just uncovering and discovering how CRISPR works and which particular proteins were involved in which aspect of the kind of the bacterial immunity discovery. And I think that these claims are directed not so much to the kind of CRISPR-Cas genomic editing embodiment or aspect of CRISPR, but more to the proteins that are involved in actually getting your CRISPR RNA and cutting your long CRISPR transcript in order to get your target RNA. And also with modifying those proteins to do things that you want them to do. So for example, this is a claim in this pending application. It talks about a variant Cy4 endoribonuclease. This Cy4 endoribonuclease, I believe, is involved in creating or splicing out your CRISPR RNA of interest. And so you could create kind of a variant of this that will then have implications on using CRISPR downstream, where you can, this variant Cy4 endoribonuclease can be enzymatically inactive in the presence of or absence of certain chemicals, imidazole, and then you can activate it in the presence of that chemical. And so you can use these variant proteins that are involved in kind of the CRISPR cascade. You can modify them to make them, switch on or switch off. And I think that this published application is directed towards that type of technology. This is a claim also in this application that's directed to a method of regulating the production of these target RNAs in a eukaryotic cell using a variant Cy4 endoribonuclease. Now, for the next, I guess, maybe 10 minutes, I think this is a kind of a terrible slide. I hate to ever put this much information on a slide, and that's why we're working on this paper. But I think that it's important to start to talk about the patents and applications that we were just talking about are in the US space. But there are a number of PCT publications that are relevant for purposes of kind of filling out the IP landscape for CRISPR. And I think that this is kind of where we're looking as we're starting to see kind of the newest publications popping up. So I'll just start by going to the first one. So the most recent PCT publication that we've identified is actually by Feng Zhang. He is the named inventor. It is out of the MIT and the Broad. They are both named applicants on this application. It just published at the end of January, and it claims a priority date back to 2012. And so I think this is kind of the first public intellectual property that we've seen coming from the MIT Broad group. And if you just take a look at Claim 1, and so again, these are not issued claims. These are the pending claims in the applications. There will be a whole series of prosecution that will take place before a patent issues. But it's interesting to look and see what these applications are actually directed to. So in Feng Zhang's application, Claim 1 relates to a non-naturally occurring or engineered Tau or CRISPR-Cas system comprising at least one switch, wherein the activity of the CRISPR-Cas system is controlled by contact with an inducer energy source as to the switch. So what we're kind of seeing, I think, most recently in terms of intellectual property that's publishing is reference to genomic editing technologies such as Tau or CRISPR-Cas, but referencing them as something that you would use in a specific type of way. And so I think this application is directed to kind of inducible control over these genetic engineering platforms. And I think if you read this PCT, it talks about being able to create the genomic editing that you want to have temporally and spatially using switches like light or chemicals or antibiotics, and enabling kind of better, more fine control over this genomic editing platform. So, these claims aren't kind of broadly directed to CRISPR-Cas9 for genomic editing. They're a little bit more specific in terms of how we can refine that technology. And again, this is, as far as we know and as of today, this is the most recent publication that has come out from any lab, and this is from the MIT and the Broad. In a slightly different direction, Sangamo Biosciences, they have a number of published applications directed to applications of CRISPR. So if you look at a PCT like this, which I think this published in January 16th of 2014, it's directed to a specific application of using CRISPR-Cas9 for genomic editing. And this particular one is directed to using it for treatment of lysosomal storage diseases, so a monogenic disease, something that you may be able to treat through kind of a single genetic manipulation. And this claim one, again, not an issued claim, but claim one of this application is directed to a method for generating a cell that produces a protein that treats lysosomal disease by integrating a transgene into the locus of that cell, such that the cell will produce a protein that treats a lysosomal storage disease. So this doesn't explicitly recite CRISPR-Cas in it. The application certainly makes reference to CRISPR-Cas, talons, and zinc fingers as ways of accomplishing this type of genomic editing treatment goal. So this is Sangamo Biosciences, if you recall earlier in the talk. They have a lot of IP directed to the zinc finger technology, and there are a number of published applications from their researchers relating to specific applications of the CRISPR technology. A second one by the same group, Sangamo, discusses a genetically modified red blood cell that comprises an integrated transgene wherein that protein either degrades or detoxifies one or more toxins or supplies a protein lacking in a lysosomal storage disease. So this is a different application, slightly different claims. Again, discusses using CRISPR-Cas9 or other genomic technologies for this type of therapeutic application. So when we gave this talk in November, the big news at that point was the publication of a PCT out of the UC Berkeley Vienna group. This is Jennifer Doudna, Emmanuel Charpentier, Martin Ginnick's group, and this PCT issued at the end of November and describes at length, 360-page PCT describing the use of CRISPR-Cas9 for genomic editing. And the claims clearly, and this is again the pending claim in that application, clearly describes kind of that technology broadly. So this claim recites a DNA targeting RNA comprising a first sequence comprising a nucleotide sequence complementary to a sequence in a target DNA. So that would be directed to kind of your guide RNA that is directed to a target in a cell, and then a second segment that interacts with a site-directed modifying polypeptide. And so again, this is a broad claim that seems to be directed to CRISPR-Cas for genomic editing purposes. It's just a pending claim. Nothing has actually happened yet in the prosecution. There has been an international search report that has cited certain references, including the Sontheimer reference that I mentioned earlier, as well as the Terns reference. not as killer prior art or anything, but as background information for purposes of prosecuting an application that contains a claim like this. And various other groups have been working on publications or published applications that relate to other applications of CRISPR. There is a published application out of Georgia Tech that actually, I don't have a claim for it, but this published application is actually directed to computing technologies for reducing off-target effects of some of these genomic editing technologies. And so this patent application is directed to using particular algorithms and computer programs to minimize the chances that your zinc fingers, your CRISPR-Cas complex will have an off-target effect in increasing the specificity and the efficiency of these technologies. This is an interesting one. So I don't know if anybody in this group has heard of Vilnius University out of Lithuania, but when we were doing our research, this PCT popped up, and this is, again, out of Vilnius University. I haven't seen any publications from that group, but they have an early PCT publication that discusses the CRISPR-Cas9 technology. And again, this is not an issued claim, but claim 1 of their application talks about a method for site-specific modification of a target DNA molecule comprising contacting under suitable conditions, a target DNA molecule, so that's your target, and then an RNA-guided DNA endonuclease comprising at least one RNA sequence and at least one of, and these are the 2 active sites of Cas9. So Cas9 makes a double-stranded break. It cuts on 2 active sites on each side of your double-stranded DNA. These are the 2 active sites of Cas9. And the result is the target DNA molecule is modified in a region that's determined by complementary binding of the RNA sequence to the target DNA molecule. And so, when we were doing our searching, we said, oh, this looks like it's describing CRISPR-Cas9 for genomic editing as well. And if you take a look, and the priority date for this particular published application is March 20, 2012, which actually predates all these others. And so, again, nothing has happened in prosecution of this published application, but it is kind of an application that's out there that is directed to CRISPR-Cas9 and appears to be specific to CRISPR-Cas9 for genomic editing. The last one that I wanted to touch on is a published application from the Fred Hutchinson Cancer Research Center. And I think that as we start to see this IP landscape develop, we're going to start to see specific applications of the CRISPR-Cas9 technology, in particular therapeutic applications. And so this is a claim, again, in a published application for the treatment of hemoglobinopathy, which is diseases associated, or diseases that would encompass, for example, sickle cell anemia, deficiencies in red blood cell capability. And this is a claim that's directed to treating such a disease comprising 1 or more endonuclease selected from a group consisting of a homing endonuclease and a CRISPR endonuclease, wherein each of said endonucleases binds to a sequence selected from, and these are a series of sequences, genomic sequences that are associated with this particular pathology. And so, again, this is kind of a more specific recitation of using the CRISPR-Cas9 system for a particular therapeutic in a particular disease. But again, it is a published PCT application with a priority date that I think right now, at least on the face of the PCT, goes back to early 2012. And so in terms of a kind of an international perspective, what's out there outside of the United States in PCT publications, there's obviously a variety of different applications directed to different applications of CRISPR. The groups that have been doing a lot of the research and have published are included within these applicants, and then there are some that not necessarily came out of the blue, but you have not been maybe talked about as much, but are certainly pursuing intellectual property that's directed to CRISPR-Cas9 for genomic editing purposes. So I think it's going to be very interesting over the next coming months to years to see how the prosecution for these applications progresses, to see what countries these applications are prosecuted in, to see what types of exchanges these applicants have with examiners about securing claims directed to some of these technologies. And I think it's something that we're going to certainly need to watch. So I mentioned earlier that I think one of the things that has been interesting about following this technology is just kind of starting to figure out what's going on, accepting that there are a lot of unknowns. I mean, the publications that we saw, those things are starting to become public, but when we started this research, we were kind of operating in a bit of a black box. But you can go and look and see what people are saying. And there certainly is a lot of talk about this technology, and organizations are actually licensing it. So this is a press release that came out in September of 2013 announcing Sage Labs having licensed CRISPR-Cas9 technology from Caribou Biosciences. Now, as far as we can tell, there's no issued patent that covers this technology. I mean, there are obviously applications out there, but there are collaborations forming. People are taking licenses and directed to this technology. And so, we kind of know without seeing exactly what is on the IP landscape, we know that it exists. The same with Horizon. Horizon licensed CRISPR gene editing technology from Harvard University. Again, we can't say what exactly that technology or what that license is directed to, what the intellectual property actually looks like. I think at this point, there's still patent applications that are not yet public, but companies are coming together and doing deals over this technology. Another one, Sigma Aldrich, sells CRISPR-Cas9 genomic editing kits. And if you look on their packaging insert, they have kind of a statement that discusses, well, if you want to purchase and use this kit, this is the associated intellectual property. And so when I looked at this, I said, oh, maybe these are the patents that are directed to CRISPR. But you go and you look at these patents and you say, oh, those are all actually directed to GFP, which I think is also incorporated into this CRISPR-Cas9 kit for particular applications. And so, they're being forthright in saying this is the IP that exists that you would need. And I think that there is CRISPR IP out there, but again, we're just starting to see it publish, and so people are purchasing CRISPR and people are using CRISPR, and the goal right now is to kind of fill out the IP landscape by watching what becomes public and how the prosecution progresses. Same thing, Life Technologies has a CRISPR nuclease vector kit that you can purchase and use. They indicate that the product or use of the product is covered by U.S. patents owned by the Regents of the University of California. Again, we don't think that we are not aware of any such patents, but I think that those patents are in the works. The same thing with System Biosciences. They also indicate that their technology is covered by patent applications, and I think we need to watch and see what these applications will ultimately issue as. But one thing I did want to point out, so there are a number of people doing research in this area. When we gave this talk back in November, Editas had just announced its launch. And it is a collaborative effort by a number of the researchers for the papers that we were discussing earlier. Among them, Feng Zhang from MIT and the Broad, George Church from Harvard, Jennifer Doudna from Howard Hughes and Berkeley, Keith Zhong from Harvard, and David Liu from Howard Hughes and Harvard University. And so, I think that the formation of this company, which is designed to kind of, I think, bring these researchers together, advance this technology, is a signal of collaboration. I think people are excited about the launch of this company. And if you go on the website, you can see a bunch of the developments that have occurred by these researchers. But again, I think the point is that to date, we don't know of any issued patents that actually exist that cover this technology. I think what we're seeing is the beginnings of published applications, and we'll have to continue to watch those work their way through prosecution. So in terms of the IP outlook and particular challenges, you can look at some of the prosecution that has taken place for some of the applications that aren't maybe directly related to CRISPR-Cas9 for genomic editing, but some of the prior work. So for example, the prosecution from the Terns application, for example, or the Sontheimer application. And you could start to get a sense of what examiners are saying about CRISPR-Cas9. Obviously, I mentioned, claims directed to isolated nucleic acids are potentially going to have Section 101 implications. The big one that we've been seeing is kind of the Section 112, the written description and enablement rejections. I think it's because it's such a new technology. A lot of examiners are saying, well, if you're reciting CRISPR, that could be any one of a million different sequences. And so this is a huge genus. It's a new technology. What we're going to need to see in these applications is, additional description in your specification, additional information to enable the invention. So kind of here, the claim that was pursued by Sontheimer at Northwestern, the one that went abandoned, was issued a final rejection on enablement grounds. And so, I think that might be something that we're going to see for some of these subsequent published PCT applications that I was discussing that are directed more specifically towards CRISPR for genomic editing purposes. And then, prior art and Section 103 issues. One of the questions that we've batted around was, well, once bacterial immunity was discovered, that principle was discovered, would it have been obvious for a person of skill in the art to use it as a genetic editing tool? And I'm sure you'll get people on both sides of the coin that say yes and that say no, but that might be a battle that's fought out in the patent office. And I think, uniquely for this particular technology, there's a number of people working on it, a number of publications out there, and I think that those publications may have prior art implications on patent prosecution in this area. There are, as I noted, for example, the Vilnius PCT. There was an international search report done on that PCT, and for example, the Doudna GINIC publication was cited as a reference in that international search report. Now, it hasn't been cited as kind of killer prior art, and we'll see how it plays out in prosecution. But I think that the existence of a lot of these publications will at least be prior art to these patent applications. Again, it may not be prior art that's going to ultimately preclude the issuance of claims, but I think it will be part of the discussion. So, just to wrap up, because I think we're over our time. in terms of conclusions for this talk, I mean, this is the second time we've given this talk. I think one of the big take-homes is that everybody's kind of watching the field. We're monitoring developments as they occur. You know, when the Feng Zhang publication came out, you know, it was like, here's the Broad, here's the MIT's IP in this area, or at least, you know, a part of it. And so it's interesting to kind of take a look at what those claims were directed to. We think that there's probably a lot more IP that will become public, and kind of the take-home is to kind of watch the field. It's unclear what the IP landscape will ultimately look like, but we're kind of putting the pieces together right now in real time. As I said, the large volume of publications may have prior art implications. You know, I ran a quick search before this talk, and right now, what we're looking at is something like 1,800 publications that, you know, relate somehow to CRISPR, of which I think about 300 of those are peer-reviewed journals. And again, that's not all stuff that's related to genomic editing, CRISPR for genomic editing purposes, but there is a lot of publication, a lot of, you know, publications in this field. And then lastly, as I noted before, it will be really interesting to watch the prosecution for some of these applications progress. We can gain some clues about how it will go based on the applications for some of the, you know, non-genomic editing patent applications that are related to CRISPR, and those may relate to things like written description, enablement, Section 101. Those are possible challenges. There's no way to tell for sure what some of these applicants will be facing, but it will be interesting to watch those prosecution endeavors unfold. And so I think we're about 10 minutes past 1. Do we want to do questions? I think I'm getting the no on questions because we're over our time. But as Pat mentioned, we have your questions, and we will endeavor to get answers to those questions to the people who asked them by electronic means. And stay tuned for our paper. All of these references and tables and everything will be included in that paper, and that will be forthcoming very soon. So thank you very much.